I'm Kirk LaPointe. Join me between 6 and 9 on Roundhouse Radio. You're listening to On Demand. Making drugs faster would, of course, save lives and money. And a new study from the University of Waterloo examines the process that actually might help us be able to do this. Chemistry professor Scott Hopkins is the author of the study. He joins me now. Good to have you on the program.
My pleasure, Kirk. Thank you for having me on your show.
We're not scientists here, Scott, so go easy with us. Try to speak in single syllable words here as we explain this. But I read about this process, and it's quite exciting, and it clearly offers opportunity. Walk us through what it is.
Okay, so in a nutshell, what we did was take very small samples of drug candidates, so on the order of nanograms. Then we isolated the individual molecules in an instrument called a differential mobility spectrometer. And once isolated, we drove very quick cycles of heating and cooling so that we could condense and evaporate water around the drug molecules. And by monitoring that process and watching how the drugs moved in this environment, we could determine things like how easily the drug could enter the bloodstream and how quickly it could pass through cell membranes. So along with that, we also used some quantum mechanical modeling to construct an atomistic picture of the drugs, and that gave us some new insights into drug properties, which we hope will help guide future drug design efforts.
So what have we been doing to date, Scott, in designing drugs?
Oh, so there's a raft of different things that are done in developmental procedures for bringing a new drug to market. Early on, it basically comes down to teams of scientists at drug companies or in academic labs who are trying to come up with particular structures that will have certain properties to target specific maladies. And typically early in the discovery process, the scientists will have quite literally thousands of drug candidate molecules that they want to try to weed out potential drugs from. And to do that, they basically try to look at the chemical and physical properties of the drugs even before it gets to clinical trials. So there's quite a bit of time and effort that goes into developing a drug. So anything that we can do to try to reduce the amount of time and resource that goes into determining the drug properties will ultimately reduce the time that it takes to bring those drugs to market and will hopefully reduce the cost to the consumer.
Where did the instinct come to focus in on this form of technology, the DMS, the differential mobility spectrometry, in order to accelerate?
So ideas like this typically come from discussions of just out of the blue science with pretty varied groups of people. So our work basically came from concepts that were bounced around between Pfizer, SciEx, the instrument company, and Waterloo. And the idea here was to try to take the smallest component, if you'd like, of a drug system, which you might have in the body or in some liquid medium, and try to understand really how that small piece of the puzzle interacted with the surrounding environment at as fine a level as we could. Then by trying to tease out the finer points, the finer details of how these species interacted, we could then try to project that towards more of an end application, a real-world application. So we're quite lucky in this sense to be able to work with industry partners who are on the cutting edge of drug development and testing, because that really is a direct pipeline to getting this out into the real world.
Yeah, sure. Scott Hopkins is my guest. Scott is a professor of chemistry at the University of Waterloo. He's the author of a study that is looking at new techniques in order to perhaps make drugs faster. and a way that would, of course, save lives because it would bring drugs into the marketplace much faster, and of course money in the way of the development of these drugs. Are you able to start to think through yet, Scott, where this can have its first applications?
Well, we're still quite early in the whole process. This is really the stepping stone off of which we'd like to build the trajectory for our drug development. What we can say right now is that we're pretty excited by the correlations that we're seeing from our measurements of these very small systems and what's actually observed in actual in vitro and in vivo measurements. So there are several different systems that we've targeted for study next, which I won't give you the details of today, but we're quite hopeful that we're going to see some really good progress over the next year or so in this regard.
Do you think that there is yet a bit of a forecast you can provide on when you might be able to start implementing a new technique like this into the development of drugs?
Well, the technique is already in place at the Global R&D site for Pfizer in Groton, Connecticut. So they are proceeding of their own accord with these kinds of measurements and working with us and SciEx to try to interpret the results and start making some more educated guesses of the directions that we can go in. In terms of projecting something like bringing a drug to market, that's certainly not in my expertise. And really, if people could forecast that, I think there'd be some pretty major bets in Vegas about what's going to happen and when. So ultimately, I think in terms of forecasting, you would have to go to the medicinal chemists at some of these drug companies to ask them where they thought things could go. From our perspective, we're pretty hopeful that this technique will start being implemented more widely, and not necessarily just in the medicinal chemistry community. There are lots of different avenues that you could potentially go down for using this technique to measure molecular properties and tease out very subtle interactions with molecules.
But when you look at the resources, the time, in a lot of ways, the trial and error that takes place in the development of a drug, have you in your own mind a bit of a goal on what a process like this might either save or make more efficient a target?
So in terms of time, the measurements that we take now, we can do in seconds, as opposed to days for comparative sort of established state-of-the-art techniques. In terms of sample size, typically running an experiment with the current technology would require milligrams to grams of drug sample, whereas we're in more of the nanogram to picogram, so millions of times less sample required. And the goal for us moving forward is to start automating our process so that we can introduce some high throughput technology and then start doing high throughput screening of lots of different drug samples. So, I mean, in terms of implementing that from a technological standpoint, that could be done in months. How that trickles down to the end goal of bringing new and improved drugs to market, there's still the high-end barrier, we'll say, of clinical trials that need to go on between the physical properties measurement and the actual selling of the drug, right? So in that regard, it's hard to predict how things are going to behave, I think.
Yeah, although it will be faster. Listen, it's been great talking to you today. A very exciting development. Congratulations.
Thank you very much.